briefly present some work that's been going on for quite a few years, which is basically working on one specific phenotypic polymorphism, which is the way a very abundant Mediterranean plant, which is the common thyme that you use for cooking, is smelling. And that sounds very innocuous, but it has actually a cascade of ecological effects. So the way the plant smells also changes the way it attracts pollinators, it changes the way it can repel herbivores, and it can also change quite dramatically the way the plant can adapt to abiotic conditions. So we've been working on that for quite a while, and we're interested in linking genetic variation with environmental condition, and also in the context where there is increasingly the wish or the belief that we can use massive amounts of genomic data, which are typically more easily acquired than phenotypic data, to sort of renew ecological genetics, where the traditional program of ecological genetics is find an adaptive phenotype, work out the genetics, and then work out how it evolves. But historically, work out the genetics meant that if the trait was not Mendelian, we would pretty much give up on it. So this is just a sort of glorious effort to try and go one level. So we're interested in genotype environment association models and how they in fine will allow plants to adapt to climate change or other things than plants. But in our case, we have, I'll come to that in a minute, where the phenotype is actually well known. There is more than one phenotype. There is actually phenotypic variation, and it's been shown by decades of field experiments and population ecology that this phenotype is adaptive, and we can actually talk about ecotypes. So the system we're working with is initially was studied as a very, very, very micro scale, conveniently placed about 10, 15 kilometers north of Montpellier, where there's been a long tradition for chemical ecology. So since the 70s, when people could do gas chromatography, they started to, in agonizing detail, describe the chemical variation in those plants. And one of the striking things that they remarked on was that in that tiny resistant, and there is more and more data corroborating that in agonizing detail with freezing experiments, etc. And then we also have a drought-resistant phenolic ecotype. So thymol is the one that you're using in cooking. You're probably familiar with the smell. Carvacrol is almost like a benzene, gasoline, like really tart smell, which is not very pleasant. And for instance, geraniol is that lemony scent, which is quite pleasant. So what was known already from work by John Thompson and colleagues at the CEF in Montpellier, the Center for Functional and Evolutionary Ecology, is that because of warming, the episodes of freezing are actually less and less frequent. So we know already that the non-phenolic ecotypes, which are the most freezing resistant, they are gradually because it was super heritable and likely oligogenic. So it was not a single locus story, but it was not going to be a hopeless hunt. At a very, very small scale, that determines most of the phenotypic variation that we're interested in. So what we did next, and that's ongoing work by Janice Erid Garcia, who's a Catalan postdoc, is cheekily put the 1,000-time partial genome project, where we're extending the idea, but instead of looking at that micro scale, we are sort of broadening up the geographical scale. And our strategy has been sort of using sort of a simple common sense rule, which is we want altitude gradients in limestone country, where we can maximize changes in altitude as a good proxy for changes in temperature regime that we are measuring with temperature loggers or using satellite data. And we want to do that in repeated transects, as many as we can. So we have Saint-Martin-de-Langres, which is the original playground, and then we have a whole bunch of other transepts. Some of them are strategically placed in good vineyards area. Others extend all the way to the Spanish border, and then all the way to Mont Ventoux, where you have the maximal altitude gradient. If you're a fan of Tour de France, that's where you'll go from around 500 meters all the way to 1,000 meters, and after that you won't have any more time. You'll only have lavender and bare rock, and a lot of people sweating on the road. Okay, so when we did that, it was in a way sort of a massive amount of data acquisition, and we came up with something reassuring and a little bit sort of boring, which was that, yes, the genotyping we had originally done mapping directly on transcriptome data, we could also do the same remapping on whole genome data, because in the meantime we had splurged on doing some packed biosequencing on one particular individual. And what we can see here is the original study playground. This is another one, which is a nearby limestone formation, where we can also see that at lower altitude we have the red dots, which phenolic ecotypes, and as we go all the way on the top of the mountain, then we'll get the non-phenolic, which are more freezing tolerant. And this is the same thing in Mont Ventoux, although in Mont Ventoux the picture is actually a bit more mixed. When we look at the amount of differentiation in the system, we can see that it's actually very weak differentiation again. So if we go from here to here, we'll have a whooping FST of 5 to 6% at most. And then we have a bunch of clusters that we can delineate using the usual structure algorithm. But it bodes well of a bunch of populations that are sort of weakly differentiated, but again, which are replicates of that ecological abiotic variation gradient that we're interested in. When we do detection of SNP phenotype associations, things are again very easy and slightly boring, which is basically we confirm that the variants that were strongly associated at that micro level scale, they remain the important variants. And so we have whooping p-values of 10 to the minus 80, which for a non-human GWAS might be a record. So we have one massive peak here, which is a SNP located in the linalool synthase enzyme, one in the terpene synthase, and one in the gamma terpene synthase. And no matter how we detect the association using principal components to correct for structure, or some more fancy and advanced kinship measures where we let relatedness be a random variable and measure in a mixed model, we find that, and we find that all the p-values are super well calibrated. Again, it's probably not a huge surprise because we're working with plants which are, again, very weakly differentiated. And again, we can see the sort of almost perfect co-segregation, at least in nature, of the ecotype and in the specific genotypes at the different. To make the story a tiny bit more complicated, those three loci, they actually are also slightly epistatic and dominant with each other. So these are three SNPs at three of those strategic locations in the genome. And then you can see here the different dominance that we've encoded. And then you can see that there is some complicated but workable patterns of epistasis and dominance in that three loci system. But to be honest, it's a three loci story, but only knowing one loci would be almost enough, because there is one loci, the one which has that gigantic low p-value here. Knowing that loci alone gives you already an R-square of circa 60 to 70%, depending on the scale at which you're working. So it's a 1 to 3 locus stories. Okay, if we can do that, then we can also make some specific simulations of the process of adaptation. We can do that, and there was a lot of work that was carried on by Kuro, who's a master's student who's just finished and unfortunately moved to Belgium to do a PhD. And we can see that essentially here we can work out what's the difference by which we respond to climate change depending on the genetic architecture of the trait. And not very surprisingly, if we have a monolocus system, we react a bit faster, and we have also simulated the one locus plus what we call two modifiers, which is the three loci that we have with the approximate R-square for the phenotypic variance we explain in the system. We can see that we can respond quite readily to that system. So, I mean, time is not endangered. We have abundant genetic variation. There is standing variation. The alleles are frequent. So we can respond quite readily to the system. So where we're heading next is that we want to incorporate more environmental data across different scales, because essentially we've been painstakingly gathering data on the ground with soil analysis and temperature loggers when they are not eaten by the wild boars. So it's pretty tedious. So we're trying to also integrate that with more remote sensing data, such as Copernicus data, which is available in greater abundance. And then we also are interested in exploring the importance of that genetic architecture for the maintenance on the longer term of that polymorphism. It turned out that the neighboring aromatic species that are also abundant in the Mediterranean, they often have the same chemotype variation. And if we do a meta-analysis of the review of where those chemical types occur around the Mediterranean basin, we tend to find the same sort of segregation with chemical types that are so-called phenolic and non-phenolic also being spatially segregated with abiotic variables explaining that quite well. The last thing we're doing also is we're trying to actually understand a bit better, and that is quite tedious, to understand the genetic basis of the variance that underlies the phenotypic variation. And then we're also doing a lot of current simulations about those genomic measures of adaptiveness and how much they are actually vulnerable to a whole bunch of assumptions, trying to use general simulation and simulations motivated by our system. So thanks.
So I'm really excited to share with you all today some work that my fantastic postdoc in the lab, Gabrielle Sandstedt, has been doing, looking at the role of heterosis in Buchera hybrids. If I can advance, which also happens every time I teach. Okay, so many of us are interested in what happens when species come together during secondary contact and form hybrids. And we're often thinking about negative epistasis, right? Kinds of interactions between genes that have been brought together or alleles at those loci that reduce fitness. So shown here is one of those classic examples, a beautiful example from Kirsten Bombley's, where she saw a hybrid necrosis. I'm too quiet? I am offended. Okay, all right. But we also have situations in which whatever. All right, subtitles. Sorry, everyone. We also have situations in which hybrid fitness is not reduced, right? So we can have compatible interactions where DMIs are not reducing fitness in hybrids. And in some cases, and we're especially interested in these cases in agriculture, we have situations where fitness is actually increased due to those genetic interactions. And so here's another classic example where we're looking at plant size in F1 maize and ear length. And obviously, ear length is going to drastically improve your yield. And so that's making more food for folks to eat. So we're super interested in heterosis in an agricultural context. So we can also call this hybrid vigor if we know the genes that underlie it. Sometimes we are thinking of this as heterozygote advantage. And many of the classic questions that we ask are, for example, what's the genetic basis? How can we maximize heterosis, right? How can we maximize that yield? And are there environmental interactions that might actually select for or against hybrids? So typically we think of dominance as being the genetic basis in this beautiful figure from this wonderful paper. If you haven't read it yet, Charlesworth and Willis, 2009, one of my faves. We're often thinking about how to maximize heterosis, and here's a study that was recently done in Kamakrista, which shows something that we see quite often, that is that F1s between highly divergent lineages are going to produce the most heterosis. And that makes a lot of sense because if you are heterozygous in certain regions, you're maximizing heterozygosity in those F1s. And environmental impacts have always been really interesting as well. So here's a study from Sheptu and Donahue, or a review, I think, or a synthesis, that was thinking about the effects of stress on outbred versus inbred lines, and how stress might reduce the fitness of inbred lines or actually reduce the fitness of outbred lines, and you can see a disparity in fitness when you compare the two. So I have some questions about heterosis. Often we see heterosis only present in some traits, and often it depends on the level of divergence between parents, the identity of parental genotypes, and the environments in which this is occurring. Now, when we're thinking about how F1s might be maximizing heterosis, what we would predict naturally from that is that the more heterozygosity you have, the higher fitness will be. And yet, studies of natural systems have actually shown us that there's very little evidence for that in the natural world. And so I was curious about if there might be regions of elevated heterozygosity that are actually underlying heterosis. And perhaps at other regions of the genome, we might see reduced fitness or no effect on fitness. All right, so the system that motivates these questions is called Buchera. Nobody actually knows how to say it. You can say it however you want, and everyone will be like, yeah, that thing. Okay, so Buchera has about 80 different members of the genus. They all diversified in less than 4 million years. They're all actually highly self-fertilizing, but they do hybridize and outcross quite a bit. And so what we actually see is that hybridization is truly disgusting in this genus. So there's well over 400 hybrid lineages representing unique species combinations that have been identified so far. And there are more to come for sure. And to show you how truly disgusting this is, right here we're focusing on one species, that green one in the middle called perennens. And it's producing all of these hybrids with all of these other green species, and they can be either diploid or polyploid, and that's the intensity of the blue there. This is a figure from Mike Wyndham, but you can imagine that all 80 species are doing this. It is truly wild. All right, so furthermore, perennens can actually outcross, and what happens with a lot of these hybrids, and I think I have it popping up here, yes, is that a lot of these hybrids or outcrossed lineages actually transition to asexuality. So hybridization and outcrossing is associated with asexual reproduction, and what that means is that you're getting these highly heterozygous genotypes and you're fixing them, right? And so this is a really cool system for thinking about hybridization and the effects of hybridization on ecologically relevant phenotypes. All right, so I'm just gonna add one last question. What can we predict about heterosis from fixed heterozygote systems? Now, this is motivated by a study from my PhD work where I planted out a bunch of plants in a reciprocal transplant, and I found that outcrossed and hybrid lineages actually had elevated overwinter survival compared to inbred sexual lineages. So the inbred sexual lineages are shown in that peach color. And I was curious about what was going on there, and I kind of thought heterosis had something to do with it. And if we actually break up that inbred group, that Peake group, into looking at lineages that were entirely homozygous or had some level of heterozygosity, we see that the heterozygous sexual lineages also have higher overwinter survival. So when I saw that, what I wanted to do was really kind of dig deep into this. So I made a bunch of F2s within this one species called Bucera retrofracta. It's the most widespread species, a lot of ecological variation, hybridizes with everyone, like disgustingly. And we've got a bunch of populations in central Idaho there, shown in blue. We generated crosses and selfed the parents, and we planted out these F2s and the parents into 3 gardens. All of the plants in one of the gardens died, so that's why you only see 2 pink points on this map here, because we kicked those out of analyses. So we planted these out in replicated randomized blocks. We had F2 lines that were created by crossing between divergent populations, so we'll call those among. And then we had F2 lines that were created by crossing within populations. So not a lot of heterozygosity within those populations. And then we had parental lines for both planted out. Gabby analyzed these data using brooms in R. And also simultaneously, this will come up later, I crossed Stricta, another model ecological system from my PhD lab, the Mitchell-Olds lab, with retrofracta, made some F2s, self the parents. I was going to put those out in the field, but they didn't like being crossed by me, so that's actually published in a different paper. But they will come up later. So we combined those field data with some GBS data, and all of the groups that I mentioned previously were included in that, along with wild collected fixed heterozygotes. So for both retro by retro, which we're calling R by R, and stricta by retro, which we'll call S by R. And you can see the full flow of work on Gabby's GitHub. She aligned these data to the version 2 of the Bucera stricta genome. There are just under 4,000 SNPs at above 5x coverage. All right, so the first thing that we found is that survival is again elevated in the F2s compared to the parents. And the fitness of within population crosses didn't differ from the selfed parents, so we just lumped those together. So we've got our within category that contains those parents, and we've got our among, and those are between population crosses. So we've got elevated survival. There's the model down below, and we included a random effect for the genetic matrix, which was generated using Plink. And then you can see the output from the model there. Now, that increase in overwinter survival did not do enough to actually make a substantial difference in total fitness. So just saying that right up front, I'm not trying to fool anyone. But there's still some other things that we could find that were super cool, which is that survival heterosis is the only trait that had heterosis, except for a weak signal in plant height, which disappeared with further ad hoc analyses. And so what we find is that we have heterosis for just one trait, survival, and that it scales with genetic distance. So what we're looking at here is survival in our 2 gardens, Caps and Mill. And you can see the selfed parents all the way over on the left with dxy of 0. And you can see, with each data point being a conditional mean for survival, that heterosis is increasing with genetic distance. All right, so then what we would predict, right, is that heterosis will be higher in crosses between more divergent species. So that's something that we predict from the literature as well. And we do see that heterosis varies with parental genotype and environment as well. So here, what we're looking at is our within F2s versus our among F2s, again in that darker blue. And what you can see is that there's just a lot of variation, right? We're looking at relative survival of our F2s compared to our parents. We have a lot of variation across genotypes, so it really matters who your parents are for producing these fitness increasing interactions. And you can see that there's one cross, which is called WI9, which has really high relative survival, but that doesn't hold up in a second garden. So this is really dependent on the environment in which you're growing them in and all of the biotic and abiotic factors that they're encountering. All right, what have we learned so far? We only see heterosis in survival, and this depends on divergence between parents, at least to some extent. This kind of opens us up to thinking about like an optimal distance hypothesis. we do see an effect of who your parental genotype is, and we do see a strong effect of the environment. Now, I next wanted to look at regions of elevated heterozygosity, and so here is a plot. We're looking at sliding window heterozygosity in our F2s, and what you can see at this is that there's two main regions of elevated heterozygosity, one at the end of chromosome 1 and one at the start of chromosome 7. So actually what we see is that these same regions are identified as high genetic diversity regions in Bucera stricta, and they're enriched for resistance genes and BLRRs, is that? Yes, okay, I got it, yeah. I can't keep track of those letters. But so this is really cool. And if, remember, go back, I had also done some stuff with stricta by retrofracta. So if we compare stricta by retrofracta F2s that I made and wild collected ones, we see the same regions popping up. But we also see that there's a little unexpected surprised dip in diversity in chromosome 5 in both wild collected and de novo stricta by retro hybrids. All right. So what we're seeing is that heterozygosity isn't uniform and that it might be beneficial in some regions and detrimental in others, which makes a lot of sense. And further, what can we predict about heterosis from systems like this, fixed heterozygote systems? First of all, sanity check. Do we see genetic intermediacy if we run a PCA? Yes, we do. Sorry for the complicated legend. There's a lot of categories of plants happening here. But if we look at admixture plots, what we actually see is that for our de novo F2s, they're right where we would expect them to be, in the middle of that triangle, and all of the wild collected stricta by retros are actually F1s. So that's super cool because we would expect heterosis to be highest in these interspecific crosses if they're F1s. It's not the same thing in the retro by retro asexuals. There's something far more complicated. So the F2s look exactly where we would expect to see them, but the wild collected crosses do not. They're kind of all over the place. And so this suggests that there's some more complex back crossing happening in these interspecific crosses. And it also suggests that the mode of asexuality is actually different when you cross within a species versus between, which is super cool. All right, so we would predict that heterosis will be higher in interspecific crosses, interspecific hybrids if they're F1s. Maybe we will see that if we tease this apart further. So this really kind of begs the question, are the benefits of heterosis driving hybridization in the wild? For some traits, hybridization will be detrimental. Can we dissect the genetic and environmental factors that are contributing to both? With that, I'll just say thank you to folks who have mentored and helped with this work, funding sources, and we are recruiting, so if you would be interested in this kind of work, please reach out.
Okay, cool. I'm gonna go ahead and start. Can people in the back hear me? Great, perfect. All right, so today I'm going to be talking about the genetic basis of complex traits in an overwintering Arabidopsis experiment. As you can see in this title slide, we have an Arabidopsis individual poking out over the snow in Toronto. So one of the main goals of ecological genetics is trying to understand how genetic diversity relates to phenotypic variation, especially phenotypic variation for ecologically important traits. So these are traits that directly affect an individual's ability to survive and reproduce. So you can think of things like nutrient acquisition mechanisms, herbivory defense mechanisms, or even reproductive traits. And then another goal in these fields is to try and understand how phenotypic variation relates to fitness differences in nature. And by understanding the relationship between genotype, phenotype, and fitness, we can then try to address long-standing questions in these fields, such as okay, all right, cool. So long-standing questions in this field, such as like, what are the number and effect sizes of adaptive loci? And then also, how does adaptation in one trait affect the phenotypic evolution in other traits? So I use Arabidopsis to try and answer these questions. Arabidopsis is a model organism in plant genomics, which means that we can take advantage of a lot of the genetic data, so genetic resources, as well as phenotypic data that's already developed by the Arabidopsis community. Not only that, but the system is great for studying ecological genetics questions because it's found across a wide geographic range, which is shown as green in this map. So that means that over the species' evolutionary history, it would have been exposed to a lot of different ecological and environmental factors that the species would have had to adapt to. So we're not the only people that are excited about genotype-phenotype associations in Arabidopsis. But a lot of these previous studies have looked at samples from a limited geographic range, so either like 1 or 2 populations or multiple populations, but within like a specific region, or they've conducted phenotyping in controlled app conditions. As much as controlled app conditions allow for controlled environments for us to conduct our experiments, they also exclude intricate environmental cues that plants will often respond to in nature. So we might not be capturing all of the genetic and phenotypic variation that we're interested when we're trying to find ecologically relevant genetic variants. All right, so for my outdoor common garden, I had 3 main goals. Firstly, to determine the effect of gene by environment interactions on phenotypic traits that we measured. Secondly, to identify, or to try and identify, novel ecologically relevant genetic variants. And then thirdly, to determine the strength of selection acting on the traits that we measured. So I used 500 accessions that were previously sampled and sequenced by the 1001 Genomes Project. Each one of these dots on the map represents a sampling location of one of the accessions that I used. For those of you that are familiar with Arabidopsis, you might know that they tend to germinate in the fall, then grow to a rosette stage, overwinter in that rosette stage, and then flower, produce fruit, and senesce in the following spring and summer. So my experiment pretty much followed this life cycle where in October, we transplanted out 6 replicates of those 500 accessions for a total of 3,000 plants. Once they were put outside, we just let nature take its course. So they got covered by snow, and then for the individuals that managed to survive winter, they then went on to flower and senesce. We measured a bunch of traits, so traits related to like phenology, morphology, growth rate, and also proxies for fitness. Today, I'm just really going to be talking about the flowering time data that we have. So we know that flowering time is ecologically important because if individuals flower too early, like this individual over here, it might produce a bolting stem that then gets damaged by the snow. Whereas all that energy that went into that bolting stem could have gone into producing a nice big rosette that might have helped it overwinter better. On the other hand, if that individual flowered too late, then it might not have had enough time to flower and produce fruit before the growing season was over. And then before I go on to looking at our flowering time data, I just wanted to show a plot of some of the environmental complexity in this common garden. This is just looking at temperature. We put eye buttons out in my common garden for the duration of it. And you can see that there's quite a lot of fluctuations here in temperature, as you might expect. And then the 2 purple dashed lines are at a constant 10 and 16 degrees Celsius. So the 500 accessions that we used from the 1000 Genomes Project, that project didn't only like just sample and sequence, they also grew these accessions up in growth chambers kept at a constant 10 and 16 degrees Celsius. So you can see that when we compare those, there is actually quite a lot of environmental complexity in my common garden, even if we just look at the single variable. All right, so looking at gene by environment interactions for flowering time, I'm going to show you a bunch of reaction plots, but I'm going to pull up one first, and then we can talk about general trends after I pull them all up. So on the y-axis, we have our phenotype, which is flowering time. On the x-axis, we have our 3 different environments, so the 10 and 16 degree growth chambers, and then my outdoor common garden. So to look if there's an effect of genotype in our environment, we just look within the single environment. So for example, over here, within this 10 degrees Celsius growth chamber data. And then if we want to see if there's an effect on environment, we then compare the phenotype data between 2 environments. So for example, the 10 and 16. We don't really see that much of an effect between the 2 growth chamber data sets, but when we compare that to my outdoor common garden, we then see this increase in slope, which indicates that we do in fact have an effective environment. Not just that, but because the lines are in parallel, that indicates that some of these accessions are responding differently to the different environments, which means that we also have an effect of gene by environment interactions. So if I pull up all 9, then some general trends that we see is we do see an effect of genotype across all 3 environments. We don't really see much of an effect of environment between the 10 and 16 degree growth chambers, but we do see an effect of environment when we compare the growth chambers to my outdoor common garden. Not just that, we also see an effect of gene-by-environment interactions, specifically in this Central Europe, Western Europe, and Germany admixture groups. Okay, and then after that, I then conducted a GWAS for flowering time. So from the outdoor common garden data, we got 5 hits, so 2 on chromosome 2, 1 on chromosome 3, and then 2 on chromosome 4, where that first hit on chromosome 4 is in close proximity to frigida, which frigida is a known flowering time gene. And then we compared, or I did a GWAS again for the 10 and 16 degrees growth chamber data, only using the accessions that we use in our outdoor common garden. And from that, we got hits on chromosome 1, but we didn't really get any hits on chromosome 2, 3, and 4. So this indicates that we get a different genetic basis when we measured it in a more ecologically realistic environment compared to the growth chambers. So that indicates that we have a SNP by environment interaction here, and that measuring these or phenotyping these individuals in a more ecologically realistic environment led us to different GWAS hits. All right, so just to go over some of the goals that I established early on in my talk. So firstly, I wanted to determine if there was an effect of gene by environment on the phenotypic traits that we measured, specifically looking at flowering time. So the reaction norm plots showed that there is an effect of genotype, environment, and gene by environment interactions. And then our different hits for our GWAS showed that there is a SNP by environment interaction too. We wanted to see if we could identify novel ecologically relevant genetic variants. We did get different GWAS hits when we did our outdoor common garden experiment compared to our, or the 1001 Genomes Project 10 and 16 degree growth chamber data. So that indicates that we might have found some new ecologically relevant variants. We're still digging into what those are. And then, as I said, this is just the flowering time data. We're going to be doing similar analyses for the rest of the phenotypes that we measured. And then the last goal that I mentioned that was to determine the strength of selection acting on these traits. So I don't have any data to show right now, but we did go out and we then collected all 3,000 individuals, and then we counted all the fruit on all of those 3,000 individuals, and there's quite a lot of variation, where some individuals had like 5 fruit and others had over 1,000. So from that data, we're then gonna use that as a proxy for fitness and use that to calculate the strength of selection, the genetic basis of fitness, and then also look for signatures of local adaptation. And then just thank you to everyone in the lab and all the help and support, and then also our funding sources. Questions? Yes.
So it seems like they really like going outside.
Yeah.
What do they not like about chambers?
I'm not really quite sure because we didn't do that experiment. And they like growing outside this time when I tried the experiment. I had tried the experiment before where they didn't like growing outside. So I think it might have had to do with this winter was a little bit more mild. So yeah.
Are the lights kind of low in the chamber?
I'm not really quite sure because we didn't do that, but I'll check. Yeah. Yes.
So in the reactioner plots, it looked like some of the regions showed really low variance among genotypes in the outdoor plots, and others showed really high.
Yeah. So.
Talk at all about that?
Yeah, sure. Well, yeah, so I guess you're looking at like North Sweden and Italy over there. So 2 things. So firstly, where we see a lot of variation in these outdoor plots, we think that that might be driven by frigida. When we looked at frigida loss of function mutations, a lot of this variation seems to be driven by frigida, which makes sense that we then got a hit for it. And then there was this one really hot week in Toronto, the week of April 16th, where everything that hadn't flowered just flowered. And I think that might be what we're seeing then in North Sweden and Italy, where they were just forced to flower really early on. Yeah. Yes.
I just have a question, because the flowering time can depend on the sowing period of the time. Did you try to sow in the outdoor condition different time?
No, so we only sowed like one time, which was in the fall, and we actually sowed them in the greenhouse and then transplanted out.
Okay.
Yeah.
And the numbers of the flowering time is at the germination or at the sowing?
At the sowing time. Yeah. We have looked at from the germination time, and it seems like we're getting similar trends. Yeah. Yes.
Do you already have just a
Yeah. So we did look, and it seems like there is a relationship between flowering time and fitness. Yeah. Yeah. Yes.
I had a question a little related to one of the earlier ones, which is how much do you think it was temperature versus how much was it day length changes? Do you think that was inducing this?
Yeah, so kind of so I can actually, maybe I can go back a few. In this plot here, this blue dashed line over here indicates the mean flowering time. So that looks like it's pretty like responsive to when we get that peak. That peak is April 16th. So it might be due to the temperature change. Yeah, but we didn't explicitly measure that, so we don't really know just yet. Yeah. Okay, cool. Great.
Hello everyone, my name is Maria Jose. I am a PhD candidate in the Quality Lab at Queen's University in Kingston, Ontario. And I'm really hoping I'm not the only one in this room who has ever indulged in some reality TV show where different contestants from around the world try to find their perfect match. And I know it's really weird to start a scientific talk talking about reality TV, but bear with me. If there's something I've really learned from those shows is that it's actually very difficult to know whether the match that is chosen is the right one. It always depends on the right place, the right time, and most of the time, all of the shows happen in a little bubble, almost like a vacuum, right? And when the contestants go out into the real world, these matches almost never work out. And that is because the real world is complicated and indeed stressful and complex. And so I'm here today to share with you how I'm trying to understand if there's an actual perfect match for natural plant populations, and how I'm really investigating the fitness and genetic effects of phenological non-random and geographical non-random matings under environmental stress. And you would think that plants are one of these organisms that have this incredible opportunity to find their perfect match that leads to fit and genetically diverse populations, especially if they're large and outcrossing populations. However, the total mating pool in a population is often not the contributor mating pool for an individual, and that is because there's variation in individual flowering schedules. So flowering across populations and among individuals can vary in its start, the duration, and the frequency, and this variation in individual flowering schedules really changes the contributing mating pool and affects mating opportunities, leading often to patterns of non-random mating. And mating systems and dispersal are fundamental determinants of population genetic structure, and they have long been recognized to be some of the primary factors that influence the rate and outcomes of evolution. And overall, non-random mating reduces the levels of genetic variation, which can speed up the response to selection. It reduces the effective population size, which leads to stronger drift and the potential for alleles being fixed in a population, which could have either beneficial or negative effects on fitness. And non-random mating can be facilitated by phenotypic differences, but also by geographic distance. But the non-random mating with respect to phenotype and with respect to geography have very different effects on the genome. So a form of phenotype non-random mating is assortative mating. And assortative mating doesn't really change allele frequencies. It just shuffles genotype frequencies, reducing heterozygosity only at the loci associated with that asserting trait, and of course any loci linked to it. Assortative mating also increases the frequency of extreme phenotypes and can create non-random associations between loci. On the other hand, you have non-random mating with respect to geography or distance, and that can actually change allele frequencies. It decreases heterozygosity and can aid in local adaptation, but can also lead to inbreeding, since there's a higher chance that, that geographically close individuals are also related. And nonetheless, the introduction of new genetic variants can also lead to outbreeding depression by the introduction of maladaptive alleles, by the disruption of co-adapted gene complexes, the introduction of hybrid incompatibilities, and genetic swamping. So right off the bat, really defining what a perfect match is difficult, and especially if there's also interactions between phenotype and geography, which you would expect happens in widespread and genetically planned species. And natural populations are not only constrained by their potential mating pool, but also by the biotic and abiotic environment. As Kathy Rochefort said, and I was going to use the exact same graph that you used, so I'm glad I used another one so that people don't have to look at the same thing twice. So work on environmentally dependent inbreeding depression has shown that the effects of non-random mating might be exacerbated under environmental stress. So in a meta-analysis done by Armbuster and Reed in 2005, they found that in 48% of the papers evaluated, fitness was significantly lower in inbred individuals under stress than in inbred individuals in a benign environment. And so the issue is that the interaction between phenology, geography, and environment in the broader context of non-random mating is still not very well understood. And so all of these things have led me to my main motivation in my research and this quest for the perfect match, or at least to really understand what are the effects of the different types of matches. So using Lythrum salicaria as the start of this show, which I will introduce in just a second, I'm really interested in first understanding how germination, growth, and fitness is affected by geography or distance-based non-random mating. Then how non-random mating patterns based on important life history and ecological traits like flowering time affect, same thing, germination, growth, and fitness. Then is non-random mating with respect to geography also affected by flowering phenology, so starting to look at interactions between geography and phenotype. And lastly, are the effects of these types of non-random mating environmentally dependent as well? So then getting all the way to a three-way interaction between geography, phenotype, and the environment. And to answer this question, as I mentioned, let me introduce you to the start of the show, purple loosestrife Lythrum salicaria. So Lythrum is it's a widespread invasive wetland perennial that is native to Eurasia and was introduced into North America initially in the late 18th century. And an interesting aspect of Lythrum is that it's an obligate outcrosser, which makes it a great system to understand these different mating patterns. And the reason why it's an outcrosser is because it has three different stylomorphs, which makes it self-incompatible. Another really interesting thing is that Lythrum has evolved along a latitudinal gradient in eastern North America as a response to differences in season length. So southern populations tend to be short. Southern populations tend to be tall and flower later because there's a longer growing season, while northern populations tend to be short and flower early because the growing season is shorter. On top of that, it's an important thing to mention, because that's what I'm going to be used as my environmental stress, is the fact that during the 90s, 2 species of beetles in the Gabrielle genus and 1 weevil in the Nanophytes genus were released as a biocontrol agent. So these beetles and their larvae feed off of the stem and the meristem, and they reduce flowering and reproduction. And to answer these questions and dissect the interactions between geography, phenology, and the environment, I'm using individuals from 20 populations collected along a 10-degree latitudinal stretch in eastern North America that have been grown in a common garden at the Queen's University Biological Station in Elgin, Ontario, since 2017. So using 2 families from each population, I perform flowering time-based crosses, sortative, intermediate, and disassortative crosses. And so I will explain a little bit the rationale between these crosses. So these crosses are based on the already known genetic differences between populations that are driven by season length. And if we look at a distribution of the flowering time, this is across a couple years, and we imagine that any individual at any given point might fall into that distribution. And let's say this pink individual flowers, this is the duration of their flowering. This individual can have an assortative mate, which is an individual that starts flowering around the same time. An intermediate mate, which is an individual that might start flowering when this focal individual is in the middle of its flowering. And a disassortative mate, which is an individual that starts flowering when this focal individual is about to stop flowering. And this is kind of when the fun part begins, and when we go out to the field and do the matchmaking. And using mating matrices based only on flowering time and the style morphology, I performed assortative, intermediate, and disassortative mates. And that's what I'm going to be called the F1 progeny for this project. So from the offspring produced from this phenology-based crosses, I then selected individuals whose parents were from the same population adjacent populations, intermediate populations, or distant populations. And that is what I'm going to be called my geographic-based families. And this really allowed me to have individuals that were progeny for both different phenotype-based and distance-based mates. I then selected 118 families from all the possible combinations of this, and germinated them in the greenhouse. And then I then selected 4 random individuals and planted them in common garden in a randomized block design at the biological station under 2 treatments. My low herbivory treatment, which I'll be calling the pesticide treatment, and a high herbivory treatment called the control treatment. I perform weekly developmental measurements in the F1s, and I'm currently also creating an F2 generation. So from all of these work and all of these crosses, what have we found so far? So the first thing is that we see quite strong effects of phenology-based matings on seed set and germination. So over here we have the mean of the relative individual seed weight. So we take the number of seeds that are in a fruit and the fruit weight to calculate the average seed weight, just because they're so tiny and there's so many, it's impossible to actually weigh all of them. And over here we have the different mate types based only on phenology. So we have assortative, intermediate, and disassortative. And we see that the progeny of assortative matings tend to have lower seed weight. And then if we look at the germination proportion between our 3 types of crosses, we also see that not only are these seeds lighter, but then also have lower germination proportion when we compare them to especially disassortative matings here, which tend to have bigger and heavier seeds and germinate at a higher proportion. So overall, we see that assortative matings produce this significantly lighter seeds and lower germination success, and this gives us an indication that assortative mating might be unmasking deleterious alleles that reduce the viability in early establishment. And in the common garden, in the first year of growth, it actually didn't show any significant differences on vegetative growth between neither the phenological, the geographical, or the environmental treatments. However, flowering phenology and reproduction were affected by geographic-based crosses and the environmental treatment. And that really reinforces how important of a life history trait flowering phenology is, especially in lithium, and it really provides initial evidence of environmental dependent breeding depression possibly. So over here we have the day of first flower in Julian days of each treatment, so the pesticide and the control treatment. So again, the pesticide is a low herbivory, the control is a high herbivory, and each of the colors represent a different parental geographical distance combination. And so we see, we found a significant interaction between, for flowering between geographic and environmental treatment. So in the pesticide treatment, we have earlier flowering than in the control treatment, but that is actually only true for the distant-based crosses. So individuals whose parents are from very distant populations tend to, in the pesticide treatment, so under low stress, they tend to flower earlier, but under high stress, they tend to flower way later. And when we look at the proportion of plants that flowered in our common garden, we see that offspring in the high herbivory treatment, so in the control treatment, had way less flowering proportion than in the pesticide treatment. And again, that's especially significant for offspring of distant parents, where they also were significantly different from individuals from the same and adjacent populations. So we see that how strong of an effect environmental stress has on reproductive fitness, but especially when they are under stress, and especially when they're just offspring of geographically distant outbred individuals. So to summarize what we found so far, we can say that finding the perfect match, it's complicated. It's always complicated. But we have really seen some interesting effects of phenology, geography, based non-random mating under environmental stress. So first, we've seen that seed set and early establishment seem to be strongly affected by phenology-based non-random mating, where assortative mating may be unmasking some deleterious alleles. We have seen that geographically distant populations, geographically distant individuals have reduced fitness, showing signs of outbreeding depression. But what I think is really, really interesting is that this signs of outbreeding depression are only visible under a high stress environment, showing that outbreeding depression could be exacerbated under environmental stress, which is something that has not been fully explored yet. Has been shown a lot in inbreeding depression, but not as much in outbreeding depression. And before I leave, I want to say that there's still lots of more to come for this project and the questions I'm trying to answer. So we're currently on the second year of growth of the F1s. We're going to try to compare also differences across years. We are genotyping the parents and the F1's offspring to also start looking at associations also with different genes. And we are also, as of right now, producing an F2 cross to really investigate the effects of non-random mating after recombination, especially because in some systems, those effects might not be shown until later generations, and hopefully we'll also get to genotype those and get some cool data out of that. So just stay tuned. And with that, I would like to thank a lot of people, and I'm happy to take any questions. Yes.
So it wasn't subsorting within those phenological classes, there were different classes.
Yeah, so they're not necessarily different crosses. Like the crosses that I do are just based on phenology.
Okay.
And then from the offspring that I get, I then create these families that are based on geography.
And do you have even representation? It's
Yes.
all just
Most-
within phenological?
Yeah. There's only a couple groups that are not completely even, but I have a pretty good representation of every single potential combination between them. Yeah. Thank you.